So with the clinical trials, it goes through different stages. It goes initially with a phase 1 approach, which is basically determining the dosage of the vaccine and getting some early readouts in terms of safety, as well as the immune response to the vaccine. That's in phase 1. In phase 2, what we look at is we're trying to expand the horizon of the objectives to include more data in terms of the safety of the vaccine, and safety is really important to us. It's important for that vaccine foremost to be safe, to be used in the population. So safety is a critical element that we actually focus on. But what we're going to be doing in South Africa is what we call really a phase 2 study, where we look at safety, but we're also going to be looking at efficacy. And what that basically means is we're going to be determining whether the vaccine protects against COVID-19 or not. So how do we actually do that? Both for safety as well as efficacy, you need to have a very similar approach, in that you take a group of people and then you randomly assign them to receive either the COVID-19 vaccine, or you assign them to receive what we call a placebo, which is almost like salt solution that is also given intramuscularly. But importantly, neither the participant nor the people that are conducting the study actually know who has received what component. The only people that will know that is a statistician and the pharmacist who is basically preparing the vaccine and would be sort of administering the vaccine. But the investigators are completely blind to who has received which vaccine, and the participants themselves don't actually know who receives which vaccine, and that is which product, whether it's a vaccine or the placebo. And that is important because we don't want to bias what happens in the study. So we don't want to have a focus on the one group that we know that received the vaccine and not focus on the other group. And also the manner in which we actually identify or go about surveying for illness is important for us to remain blinded. So in the South African study, we've got 3 components to the vaccine study. The first group are people without HIV, and it's a small group of 50 individuals. And we're going to investigate in that 50 individuals whether 1 dose of vaccine or 2 doses of vaccines are required. So we don't know which is going to be the optimal dosage schedule in South Africa that will induce the best immune response. So that's the first group, and half of them will receive the vaccine, the other half will receive a placebo. In the second group, again, it's limited to people with HIV, and this is a much larger group. This is about 1,900 people now who will get a single dose of the vaccine. But if we show in the first group that you need 2 doses of vaccine, the second group will be adapted to include 2 doses. But the starting point for the second group is to receive 1 dose of vaccine in 1,900 individuals, half of who receive the vaccine and half of who receive the placebo. These individuals will be followed up to look at how safe the vaccine is when it's given to them, and that follow-up continues until 1 year after they were vaccinated. In addition, we'll look at the immune response to the vaccine, and throughout that 1 year period, what we'll do is we'll take regular swabs from them. Every time they experience a respiratory illness, when they have a cough, a sore throat as an example, a headache, they will need to come back to the clinic, or we will need to go to their homes to actually get a swab. Right? And that swab is then tested, a nasal swab, and that nasal swab is then tested to see whether or not they might have been infected with the virus. So that is the efficacy part of this 1,900 subjects. We're wanting to see whether the vaccine is safe, whether the vaccine is immunogenic, and whether the vaccine is efficacious, which means we're wanting to see whether there's going to be any difference in terms of clinical illness, which is what we call COVID-19, between the group that received the vaccine and the group that received the placebo. And then the third group is another small group of 50 individuals, which are people that are living with HIV now. And the reason why it's especially important to investigate the group in the South African context, as we know, is that South Africa has got the highest prevalence of HIV in the entire world. Roughly about 14 to 15% of our adult population are living with HIV. Right now, we're not too sure whether people living with HIV are at increased risk of developing severe COVID-19. There's some indication that they might be at a higher risk of developing severe COVID-19. But immaterial of that, we need to know how well people living with HIV actually mount an immune response to this vaccine, as well as what is the safety of this vaccine in people who are living with HIV. So we're planning on doing another small group of individuals, 50 HIV people living with HIV, 50 individuals that are living with HIV, half of whom will receive the vaccine, half of whom will receive the placebo. And then we'll look at a safety profile in those individuals, as well as how well they mount an immune response. And if we find that the type of immune response they're mounting is similar to people without HIV, then we wouldn't need to do another large study to determine vaccine efficacy, because if the immune response is similar, we would expect the efficacy to be similar. But it's important to get that information early on so we're not delayed when, if the time comes that we show the vaccine to be efficacious and we're wanting to introduce it into South Africa, into any other country, we don't delay its introduction in the population that are living with HIV because we don't have evidence as to whether the vaccine is going to work in that group or not.